Okay, good afternoon. Now we are going to start with a new block number 2. This will be mainly about the nervous system and the endocrine system, and you will find that these two systems are so interrelated with each other. Like by probably the end of the unit, we will have some comparison between the nervous system and the endocrine system because they are related to each other. They communicate with other cells by using messengers, for example. These messengers could be either locally secreted, like in the neurotransmitters in the nervous system, or they could be circulating in the blood and affect many other cells in the blood, like in case of the endocrine system. So there are some similarities between the nervous system and endocrine system. They are not separated from each other. So this will make this unit like one complete block. So as we started with any other system before, we start with the histology. And today we are going to deal with the histology of nervous tissue. Remember the 4 types of basic tissues that we have? We studied the epithelium, we studied the connective tissue, studied the special type of connective tissue, and then we studied the muscular tissue, number 3. And now today, number 4 is we are going to study the nervous tissue. First of all, let's have a brief introduction to the functions of the nervous tissue. This is just a very simplified idea, is that it controls and integrates all body activities, as I said, along with the endocrine system. And there are 3 basic functions. If you can compare them like to the functions of a computer, they are input, processing, and output. So this is probably the same. Input is the sensory input. The receptors are going to detect the environment, whether inside the body or outside the body. Like, for example, I'm detecting that there is light that is outside the body, but at the same time, I have a colic in my intestine, so that is inside my body. So you detect inside and outside stimuli, and then that will be integrated, like in the central processing unit of the computer. There's integration, interpreting, remembering changes, and taking decisions. What are we going to do? And then after that, there is the output in the form of motor activity. This motor activity is not necessarily an action of muscle. It could be muscle, whether voluntary, involuntary, or cardiac muscle, or it could be a gland. So in a gland, the gland will secrete, and this is what we call a secretomotor stimulus. So it could be motor or secretomotor. It affects both muscles and glands. The divisions of the nervous system, we have structural and functional divisions of the nervous system, so please don't mix them together. Structurally speaking, the nervous system consists of a central nervous system and peripheral nervous system. And the central nervous system, as you can see, is the brain and the spinal cord. It is the part of the nervous system that is hidden inside the bones, whether inside the skull or the vertebral column. And then we have the peripheral nervous system, and this consists of cranial nerves and spinal nerves. So the nerves that are connected to the brain and arise from the cranium are the cranial nerves, and we have 12 pairs of these cranial nerves. We're going to have an idea about them later on. And the nerves that are connected to the spinal cord are therefore spinal nerves, and they arise from the vertebral column through the intervertebral foramina. Both of these types of nerves, because we have 12 pairs of cranial nerves, we have 31 pairs of spinal nerves, both of them, they carry sensory and motor function. So some of the nerves are sensory, some of them are motor, purely motor, but many of them are mixed together. They have sensory and motor function. The sensory is called also afferent, and the motor is called efferent. So afferent and efferent. Efferent, motor, E for exit. So the impulses exit from the central nervous system. That's the efferent. Okay? So they transmit the impulses from the central nervous system to muscles and glands, to the effector organs, or effector structures, not necessarily organs. So this is the structural division of the nervous system, central and peripheral. Now, functionally speaking, the nervous system could be somatic or voluntary, or it could be visceral, which is the involuntary part. Okay, so like somatic, motor, it has motor and sensory. Each one of them has motor and sensory. So the motor, it transmits messages from the central nervous system to the skeletal muscle, voluntary muscle. So this is the, we call it somatic motor neuron. It affects the muscles that are under our control. If we look at the motor part of the visceral nervous system, which is the autonomic nervous system here, you will see that it transmits messages that go to other type of muscles, to smooth muscle and cardiac muscle, the involuntary muscle. So they increase the heart rate, for example. They cause erector pylorem, they cause contraction of erector pylorem, let's say, which are located in the dermis. They cause secretion of the glands, like, for example, sweaty glands start secreting, if you are in fear, for example. So this is the motor part of the autonomic. If we go back to the somatic, the sensory part, it transmits information from external environment, information that are mainly detected by skin. We call them tactile sensation, like pain, touch, temperature, pressure. Okay, all these can be detected on the skin. We are going to study the receptors. Each one of them has special receptors, or sometimes receptors can carry more than one sensation. Or special sensations, like you are hearing me now. This is an external environment, hearing, smelling, vision. All of these are somatic sensory impulses, and they are produced in the external environment. In the internal environment, as I said, that sensory impulses transmit information from the internal environment in the visceral nervous system. In the visceral nervous system, sensory neurons transmit information from the internal environment to the central nervous system. Like, for example, as I just mentioned, you feel sometimes acidity in your stomach. Okay, distension, bloating. You feel that, for example, pain that arises, for example, from the heart because of reduced blood flow, which is called angina or myocardial infarction. All these are visceral sensations. They are not somatic sensations. Like, for example, let's say if you are conscious and somebody opens your abdomen and took out the stomach and touches your stomach, you will not feel it. But if there is gas inside your stomach, which causes distension, that will give you feeling. So these somatic sensations, they have different quality from the visceral sensations. So this is what I'm saying, functional divisions. We have either somatic nervous system or the visceral nervous system, which is, both of them are either motor and sensory. And with the visceral, there is something else that nowadays they are concentrating more on. They call it enteric nervous system. They found that there is a very complicated network of neurons, ganglia, in addition to glands, that are present in the gastrointestinal tract. And they can control the function of the gastrointestinal tract irrespective of the brain or the spinal cord. So they can work on their own, but they can be modified by the brain. Like, for example, let's say there is one reflex that probably many of you has experienced. If you wake up in the morning and you have a glass of water, then within 2 minutes probably you will feel the urge to go to the washroom. This is what we call gastrocolic reflex. There's not enough time that what you have put in your stomach will go to your large intestine, but this is a reflex activity that the stomach has felt something, and the enteric nervous system has processed that and gave orders to the rectum to start moving the muscles and peristalsis and have defecation. And then if you want to go back to sleep, then you still have control and you can use your brain and stop it. Use the sphincters, which are the voluntary sphincter, and stop that. Okay, so as I mentioned, the enteric nervous system, it can control the local environment inside the gastrointestinal tract, but is modified or can be modified by the central nervous system. Going back to the visceral, probably we mentioned that before, and I will mention it now again, and we're going to have a full lecture on that, is that these motor neurons, they could be sympathetic or parasympathetic. So the autonomic nervous system is in two parts. It is the sympathetic, which is for fight or flight, and the parasympathetic, which is for rest and digest. So again, like for example, let's say sympathetic nervous system, when it is stimulated, it will cause secretion of glands. But which glands? Like glands in the skin, sweaty glands. But it will not cause secretion of glands in the stomach, because these glands are for rest and digest. So it is the parasympathetic nervous system who's going to work on them in order to do rest and digest. And also the cardiac muscle fibers, they are stimulated to, although they have their autorhythmicity, but they are stimulated to increase their rate by the sympathetic nervous system, and the parasympathetic nervous system will depress them down. Smooth muscles in the wall of the gut, they are stimulated by the parasympathetic nervous system to transmit the contents from one place to another, rest and digest. But at the same time, there is another group of smooth muscle fibers in the gastrointestinal tract, we are going to talk about this later on, that form sphincters, like valves between one region or another. So these muscles, they should be relaxed. So it's not necessarily all the time it's a stimulation, it's sometimes stimulation and inhibition by the same component of the autonomic nervous system in order to serve the function, okay? So probably this is more functional introduction, but it will give you an idea about the structures that we are going to talk about later on. Here you can see that there is a link of an animation. Probably this is the second time or the first time you have such a link, but you will see many of these links. This is a link, an animation by Wiley Plus. So if you are connected to the internet, and you can click on it and go and watch the link about the functions of the nervous system. Now, let's go with the actual histology of the nervous tissue. So the nervous tissue, there are two main types of cells. There are the neurons, which are the specialized cells. They are the functional cells of the nervous system, the specialized functions. They are the excitable cells that transmit impulses, and there are the neuroglia. The neuroglia, the term neuroglia means, neuro means nerve, and glia means like glue. So they are, the first anatomists thought that they are glue for the nervous system, and in fact they are glue for the nervous system because they support. They are the supporting cells. They provide nutrition for the neurons, and they protect the neurons. So they are like the connective tissue of the nervous system, okay? Neuroglia. Their number, more than the number of the neurons, but they are smaller than the neurons. So they occupy about half of the volume of the central nervous system, but because they are smaller, then their number, they outnumber the actual neurons. They differ from neurons in that they are not excitable, of course, as I mentioned, but they can divide. A neuron cannot divide. It's not like the epithelium that can divide. It's not like the bone that can, the osteoprogenitor cells can divide. Here, neurons, they cannot divide. Remember, like for example, cardiac muscle fibers, they cannot divide. So these neurons, they cannot divide when they are mature. So it's the neuroglial cells that divide. That's why when we have tumor, it's usually a tumor in the glial cells. So they have like gliomas. It's a tumor arising from the glial cells. There are 4 types of these glial cells in the central nervous system. Again, to remind you, the central nervous system is the brain and spinal cord, and there are 2 others in the peripheral nervous system. So all in all, we have 6 glial cells. These are the names of them: the astrocytes, oligodendrocytes, microglia, and epyndemal cells in the central nervous system, and Schwann cells and satellite cells are located in the peripheral nervous system. Let's deal with each one of them separately. Astrocytes, as the name indicates, astro means like star, like in astronomy, astrology. So astrocytes, they are star-shaped, and you can see they have multiple processes and they are star-shaped cells. They are important for maintaining what we call the blood-brain barrier, BBB. So this blood-brain barrier is a protective for the brain tissue, because the brain tissue is so delicate, and the environment should be kept in certain ranges of pH, electrolytes. So it's very delicate. So that's why there is a barrier that selectively allows substances to pass into the nervous tissue and to prevent others from passing between blood and the brain. That's why it's called blood-brain barrier. And this blood-brain barrier is mainly produced by the junctions that are present between the endothelial cells. Remember, we have, like, these are endothelial cells here. Here's the blood. And endothelial cells, they have like tight junctions here in between them, so they prevent the passage of substances in between the cells. So this is the main component of the blood-brain barrier, but the astrocytes, they contribute. They are not the main component. They contribute to the formation of the blood-brain barrier. In fact, they will stimulate these endothelial lining of the capillaries to produce more and more of these tight junctions to make the blood-brain barrier unpenetrable. So, as I said, they maintain this appropriate chemical environment. They take up excess neurotransmitters, regulate potassium balance, and as you can see here, they have multiple processes. So they act like a conduit between the blood vessel and the neurons. So the nutrients will pass from the blood vessel to one of these processes, to an astrocyte, and then will be delivered to the other neuron, as well as their general function as a whole, as neuroglial cells. It is the support. So they provide structural support, they provide nutrients, and they enhance the blood-brain barrier. The other cells are called oligodendrocytes. Oligo means less. Like, for example, oligospermia, a person who has few sperms in his seminal fluid. So oligo means less. Oligodendrocytes, these cells, they have less processes. And they are responsible, you can see them here, they are responsible for the formation of the myelin sheath in the central nervous system. So they make a wrap around the processes of the neurons. This is called the sheath, the myelin sheath. Okay, and this wrap is important. At least it acts as an insulator. The second function is that it increases the conduction velocity of that process. So in the central nervous system, the cell that is responsible for that myelin sheath, formation of that myelin sheath, is the oligodendrocyte. In the peripheral nervous system, there is another cell which is called Schwann cell. It's another glial cell. Remember, if you go back to this slide, I said that there are two types of glial cells in the peripheral nervous system, Schwann and satellite. So now we know which cells produce the myelin sheath: oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. The other cells are called microglia. Micro means small, and this is a description of their size. Microglia. They are very small cells, and they are derived actually from bone marrow. They are like macrophages. Okay, so they digest like debris, dead cells, any foreign material. So they are the microglia, phagocytic cells. Then we have the epyndemal cells, and these are, as you can see, they are like cuboidal or columnar cells. They line the ventricles of the brain. We're going to talk about this probably next week, that inside the brain, there's a ventricular system. There are spaces, and these spaces are washed and contain fluid, which is called cerebrospinal fluid. And these ventricles continue into the spinal cord as a narrow canal called the spinal canal. So this fluid should be secreted. And these are the epyndemal cells that secrete the cerebrospinal fluid because they line this ventricular system. They are present in its lining, therefore they can secrete it. And they have cilia, they have microvilli as well, these epyndemal cells. The other types of cells are present in the peripheral nervous system, and the one here is called satellite cell. Can you see them here? Like this is a neuron. You see, it's a very big cell. I just mentioned that the nervous tissue cells are big, but the glial cells, they are small. All these surrounding it are glial cells, They are satellite cells, and they are present in the peripheral nervous system. So in the peripheral nervous system, we have some collections of neurons in the peripheral nervous system called ganglia. We are going to talk about that. There's a dorsal root ganglia, there are sympathetic ganglia, parasympathetic ganglia. These are small collections of nerve cell bodies, and they are surrounded by these satellite cells for protection and support again. And the second type, now you are familiar with the Schwann cell. This is the second type of cell that is present in the peripheral nervous system and is responsible for producing this wrapping material, the myelin sheath, for the processes of the neurons. And of course, we will see that probably in the last slide, that they participate in regeneration of a damaged axon. We will see that later on, but they are very important, in fact. So far about the glial cells. Now let's deal with the neurons. So the neuron is the functional unit, as we mentioned, of the nervous system. They are excitable. Okay, in physiological terms, they produce action potential, and they have a cell body. This is the cell body, or it's called perikaryon. The other name is called perikaryon, and you can see that it has a single nucleus with a very prominent nucleolus. Presence of nucleolus usually indicates that there is good amount of protein synthesis. Not only that, there's another indication here that they have what is called Nissl bodies. There are multiple dots called Nissl bodies. In fact, these are rough endoplasmic reticulum as we see them under the light microscope. And as you know, that the presence of rough endoplasmic reticulum is a good indication that there is a considerable amount of protein synthesis. A lot of proteins are required here to produce the microfilaments, microtubules, and participate in the formation of the neurotransmitters. So there are a lot of neurofibrils, okay, like intermediate and microfibrils. These are important for support. Remember these when we dealt with them in probably the second lecture. And then there are microtubules, small tubules that help, and they are all made of proteins, like tubulin. Remember the protein tubulin, and they transmit substances within the cell. You can imagine here that this cell, like some of these cells, probably they are more than 1 meter in length. Some of them. They have the perikaryon, and then there's a very long axon, and you have to transmit a lot of substances through that axon, which is going to form the nerve. Like for example, we have a nerve that its cell body is located in the lumbar region here in the spinal cord, and then that nerve continues all the way down to reach the big toe. So you can just imagine, okay, the length of that cell. Not all the cells are that length or that long, but just to give you an idea about, there are many such cells. They are very long. They require a lot of microtubules to transmit these substances, and they have the processes. So these processes, I'm sure that this is like when I'm going to talk about the processes, it will be like high school biology. It's processes. We have the dendrites and the axons. Usually we have multiple dendrites, as you can see here. The dendrites are the receptive zone. They conduct impulses toward the cell body, toward the perikaryon, as you can see here, the direction of the arrows. And they are short, branched. They are unmyelinated. They don't need to have that wrapped, the myelin sheath. And they have many specializations here for synapses to connect with other cells. Like one cell, there's another axon might come in here and connect with these from other cells. Sometimes it's another dendrite, but usually it is from another axon. And again, they contain neurofibrils and Nissl substance. The axon, usually it is a single axon, as you can see. It's very long and thin. It is cylindrical process. Sometimes it has a collateral, another branch that runs with it. But all the time, the distal end here will divide into very small branches, and these branches descend at their end. They are called synaptic botons or synaptic bulbs because they look like the bulb of an onion. So they are distended like that at the end here. And they carry impulses away from the body of the cell. They start here at this location, which is called the axon hillock. This is a hillock, means a small hill, so probably it looks like a small hill. So that's why they call it the axon hillock. And then there's here an initial segment. At the initial segment, the decision will be taken, because these dendrites will receive, sometimes they will receive excitatory and sometimes they will receive inhibitory signals. So it depends on the balance between excitatory and inhibitory signals that are received by these dendrites, that this initial segment will decide whether to trigger an impulse or not. Probably we'll deal with more details about this from the physiology. I'm more concerned about structure rather than function. And these axons, while the dendrites are unmyelinated, the axons, they might be myelinated. Some of them are myelinated, some of them are non-myelinated. The myelinated ones, as you will study that in the physiology as well, the myelinated ones are usually the rapid ones, the rapid conducting ones. The ones with the RAP, they are the rapid conducting ones. So these are the myelinated axons. Axons are surrounded by a myelin sheath, which is a multi-layered lipid and protein. And this myelin sheath is produced by Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. Probably now you are wondering, what is this myelin sheath you are talking about? So let's say we have here a section of an axon, and this axon will be surrounded by a cell, which is a Schwann cell. That is the nucleus of a Schwann cell. What happens is that the Schwann cell will start wrapping itself around the axon. One wrap, second wrap, third wrap. Now I'll draw it here. That's the axon, and that's the Schwann cell. It's wrapping itself. While it's wrapping itself around, it is squeezing the cytoplasm of the cell out. So these wraps are purely cell membrane. Cell membrane, cell membrane, cell membrane, one over the other. And as the cell is wrapping itself around, the cytoplasm is squeezed out. So all this wrap is made of cell membrane. What is the main component of the cell membrane? Lipids. And lipids, phospholipids, and proteins. So mainly it is lipids, in fact, and proteins, as well as carbohydrates. But we're talking about lipids. So multiple wraps, we have a lot of lipids here. And then at the end, after all these wraps, we will have the cell and the nucleus sitting here. And inside is all lipids. So that's why this will creates what is called in the nervous system, it's called the white matter. The white matter, it looks yellowish white because there is a lot of lipid in it. The lipid that wraps around, and at the same time it works as an insulator. And because these Schwann cells, you can see them here, for example, they are not continuous with each other. There is a gap in between them called node of Ranvier. Then the electrical impulse will not be able to move directly through the lipids. It will jump from one node of Ranvier to another node of Ranvier to another node of Ranvier. It will leap, it will jump. Therefore, the transmission will be faster because it's jumping. It's not just moving from throughout the entire length of the axon. Okay, so this is myelination. Myelination in the peripheral nervous system is produced by Schwann cells. Similarly, in the central nervous system, there's myelin sheaths. There are axons that are myelinated, but we don't have Schwann cells in the central nervous system. We have the oligodendrocytes. So these oligodendrocytes, with their processes, they will send one process around one axon, another process around the other axon, and another process around another axon. In other words, one oligodendrocyte participates in the formation of myelin sheath of more than one axon. One cell participates in myelinating more than one axon. But if you look at it here, regarding the Schwann cell in the upper right corner, you will find that one Schwann cell myelinates one axon. doesn't participate in myelinating the other. So this is the difference between the myelin sheath in the central and peripheral nervous system. These are the processes of oligodendrocytes. This is one process myelinating this axon, and another process myelinating another axon. But here only one cell wraps around one axon in the peripheral nervous system. Okay? You will find all these details on the other slides. They are not mentioned here, but they are definitely mentioned in the other slides. So it appears white, hence it's the white matter. We call it the white matter. But the areas that contain nerve cell bodies and unmyelinated axons, they will appear gray. That's why it's called gray matter. So in the brain, there's white matter and gray matter. The gray matter is composed of nerve cell bodies, dendrites, because dendrites are all unmyelinated, and unmyelinated axons, while the white matter consists of myelinated axons. And if these axons are destroyed in one type of diseases here, it's called multiple sclerosis. Probably you have heard about it. Multiple sclerosis. This will cause destruction of the myelin sheath. This will definitely affect the conduction velocity to start with, and at the same time it will produce short circuits because the insulator is damaged. So there is short, like when you have two electrical wires, you bring them close together, so you'll have like short circuits. That's what will happen. There's no insulation. This is what I have just mentioned. Like there are spaces, gaps between the myelin sheath called nodes of Ranvier, and the conduction in myelinated axons is, we call it saltatory conduction. There's leaping of impulse from one node to another. This is, in unmyelinated axons, it is continuous, not saltatory. And the continuous is slower than the saltatory. So some of the axons are myelinated, others are unmyelinated. In unmyelinated, it doesn't mean that they are not surrounded by a cell to act as an insulator. Okay, like it's like the electrical wire. You should have insulator. You should have something plastic around it. Okay, but not necessarily a thick one. So as you can see here, like this is an unmyelinated axon. It's a Schwann cell just hugging or surrounding multiple axons, but it is not wrapping itself in multiple layers around it. So there is no myelin sheath. So, and the impulse can pass continuously along the surface or the membrane of the axon, the cell membrane of the axon. So it's the same thing in the peripheral central nervous system. The unmyelinated axons are just touched or hugged or covered, not wrapped by the processes of oligodendrocytes instead of Schwann cells in the peripheral nervous system. This is to show you the myelination process. The myelination process is a continuous process. It continues actually during the first year of life as well. So this is then Schwann cell, and as you can see, it started wrapping itself, squeezing the cytoplasm out, and this is all the cytoplasm. There is no pink material here. All has been squeezed out. This is the myelin sheath. It is wrapping itself around. And as I said, this process continues throughout the first year of life. That's why in newborns and in babies, you will find that their responses to stimuli are not as rapid or not as coordinated as in the adults, okay? Because the impulses are slower. The myelin sheath has not completely developed, okay? And this is again to show you, this is more of a physiology slide rather than an anatomy, but just again to give you an idea about saltatory conduction in myelinated axon and a continuous conduction in non-myelinated axon. Here we have the synapse. As I said, that the terminal part of the axon is called the button or the synaptic bulb. So it's like a bulb, it distends, and these are the secretory vesicles. They contain neurotransmitter. Neurotransmitter or secretory vesicles reach this surface of the cell and they open, And so the neurotransmitter will be in a gap. Here there is no direct contact. This is a gap, a space. Extracellular fluid is located here. And just opposite to it, here, for example, we have another neuron. It's not a neuromuscular junction. It's another neuron. And here there are receptors for that. Can you see the proteins, the green ones? The proteins here. These are receptors for the neurotransmitter. So they will stimulate the next neuron to be excited. Okay? And then there are enzymes that are going to destroy the neurotransmitter, and another neurotransmitter will be there, and so on. So structurally speaking, this is what we call a synapse. There is a presynaptic neuron, this one, and there is a postsynaptic neuron here, and in between them there is a synaptic cleft. The cleft is a gap. They do not touch each other. There are neurotransmitters that have to swim until they reach the other cells. This is to show you a structural classification of the neurons. So most of the neurons are multipolar. That's to say they have multiple dendrites and usually a single axon. So these are most of the neurons. Some of them are bipolar. So as the name indicates, they have two processes. One of them will work like an axon and one will work like a dendrite. You will find many of these when we are going to describe the sensory organ, for example, in the ear, in the retina, bipolar cells of the retina, for example, in the olfactory epithelium. So they are present in those areas. The other type of cells, which is a little bit complicated, when most of them they are sensory neurons. You can see that it is like unipolar. So there is a perikaryon here, and then connected to it is only one process. But this is a very short stem of the process. Immediately, it will divide in like a capital T shape, okay, into one process which will go peripherally. So it will act like a dendrite, and the other goes centrally. So that's why these cells are called pseudo-unipolar. They are unipolar, but they are not actually unipolar. The unipolar is just like probably few micrometers, and then it divides. So they are called pseudo-unipolar neurons. And they are present, as we will discuss that later on when we will deal with the spinal cord, in dorsal root ganglia. Dorsal root ganglia. A ganglion is a collection of nerve cell bodies outside the central nervous system. No worries, I'm going to go back through all these terms that I mentioned right now, the pseudo-unipolar neurons, the ganglion. I'm going to go back and deal with them because they are very important to understand the formation of a spinal nerve, which we will have next time. Functionally speaking, neurons are either sensory or motor, or they are interneurons. They are just connecting, integrating. They are neither sensory nor motor. This is to show you about that the neurons have a special architectural circuits. This is a science by itself, actually. I just wanted to read it and have an idea about it, just an introduction. Some of these circuits, it's like the computer circuits now that they are designing for artificial intelligence. It's something like that. So this is actually what we have in our body. Like this is a converging circuit, multiple neurons affecting one, or this is a diverging. It's one neuron that's being amplified because it affects two, and then the two affect four. So there is amplification, or there is reverberating. The last neuron will stimulate the first neuron to produce more impulses, and so on. So these are the types of neuronal circuit: diverging, converging, reverberating, and parallel after discharge. This is the last topic today. It's about regeneration or repair. Now, the thing that you should keep in mind is that if the nerve cell body is destroyed, then there is no regeneration, no mitosis, no regeneration, and the other neurons cannot multiply, okay? So we are talking about damage of the processes. If the processes are damaged, then there is a possibility that the processes will regenerate if they are in the peripheral nervous system. But if the injury is in the central nervous system, then this issue is still under investigation, and there is little hope that this will happen. That's why it's still a problem that if you have, like if the patient have a spinal cord injury, transection of the spinal cord, while if a nerve outside here in the hand is cut and the nerve is cut, then there's a high possibility that the axons of that nerve will regenerate. But if the spinal cord, central nervous system, is affected, then regeneration is very little and does not, even though that the neuron, the nerve cell body, is still viable. What we have, like what they do for physiotherapy sometimes, is that there is some sort of plasticity. That's to say, these circuits or these neurons, they will change they will have new processes, they have new connections, they have new proteins, and they will make new circuits, okay, in order to bypass the deficit. So that is not regeneration, that is what we call plasticity. So sprouting of new dendrites, synthesis of new proteins, and producing new synaptic contacts. This is what happens if a nerve is cut in the peripheral nervous system. So the nerve can regenerate, but we have to know that the cell body should be intact. Schwann cells should remain intact. Remember, we mentioned that Schwann cells are important for regeneration in that previous slide. So that's why, because the Schwann cells are going to regenerate and produce a tunnel, a new tunnel for the sprouting axon to pass through. If we don't have these Schwann cells, then the axon will lose its way. Okay, so that's why those Schwann cells are important. And as we know, that Schwann cells are glial cells. They can multiply. Okay, so they multiply and produce and provide the new tunnel. So we have to have a neuron cell body remain intact, Schwann cell remain active, and scar tissue does not form too rapidly. So that's why sometimes if they just suture the wound and don't take care of let's say there was a nerve injury, then there will be scar tissue in between the two pieces of nerve, and nerve cannot grow. So that's why they have to bring the two pieces together, approximate them, so in order not to produce scar tissue, connective tissue that obstruct the sprouting axons. And this is what's going to happen. Like after the injury, there will be chromatolysis. Nissl bodies will break into small granules. Then the remaining part of the axon will disintegrate distal to the site of the injury, so that the same is true for the Schwann cells. And then the Schwann cells will multiply again, form a tunnel, and the nerve will grow with a speed of 1.5 millimeter per day.